Hello, everyone. It's time for something new and exciting, genetics. Let's take a look. It's going to be something different. It's actually a review of stuff that you already probably know. And if you don't remember or you don't know, well, then this is going to just help you. So what is genetics and what is heredity? So genetics is the study of heredity. Wow, thanks. That really helps. And how traits are inherited. So let me tell you what heredity is before you get mad. Heredity is how traits are passed from parent to offspring. So genetics is just studying why do children look like their parents, and why does sometimes they look like one parent, and why does sometimes they look like another, or why is it sometimes they don't really look like either one, not totally. Or maybe somebody has blue eyes and no one else in the family has blue eyes, so are they adopted? No. There's so much stuff we could talk about, like Gregor Mendel, who was really the father of modern genetics. But we're not. We're actually going to talk about Punnett Squares today, and that's what this video is about. How do I read a Punnett Square? How do I look at it? How do I know what it says? What goes on the outside? What are these weird words? Genotype, phenotype, dominant, recessive, what do they mean? So we're just going to be talking about basic Punnett Squares today. We're not going to be talking about anything advanced. We're not going to be talking about anything other than complete dominance. Those are the basic Punnett Squares that you might be used to, that you might have done already, or that you might have done in class. So we're going to take a look. We're going to cross these two parents. One has a big P and a little p. The other one has a big P and a little p. These letters together, we call that the genotype. The genotype you can think of as what alleles or what genes that creature has. In this case, we're talking about a flower. The capital denotes or shows that it's dominant. Dominant is the trait that you readily see. If you think of a dominant person, they're maybe more present in the room. You would notice them right away. They take control or take charge. So a dominant trait kind of takes charge. And recessive, if something's recessed, it's kind of moved to the back. I think of it more as like passive, I think might be also a good word for that, where it's just like, you know what, dominant trait, you can go ahead, you can show up. That's totally fine. I'll just be here in the background. Don't mind me. Don't pay attention to me. I'm just here. But if there are two recessive traits, so if we had two little p's here, then we would see the recessive trait. So if we had two recessive alleles, that's two little p's. It could be little r, it could be little t, it could be little whatever letter. It could be little lambda, I don't know. If we had two little letters, then we would see that recessive trait. In this case, since we see a big P and a big P on both of them, we know we're going to see the dominant trait. So our genotype is big P, little p, that's the letters. And the phenotype, that's the physical trait that we can see with our eye. So you can know my phenotype for things. Like I've got, I guess, like blue eyes. So that's my phenotype. But you don't know my genotype. That's what genes and genetics I have. I don't know. I really don't know. So when we make a Punnett square, we put the genotype, the letters of the parents, on the outside. Here's one parent. A lot of times they put the male on the top and the female on the side. Why? I don't know. They just do. Okay. Do you have to do it that way? No. If you don't want, I guess. It doesn't really totally matter, but that's just usually how we do it conventionally. Same thing, we usually put the capital letter first. Just like in your name, the capital's first. We put the other parent on the side. So these are the genotypes of the parents. Well, what's a Punnett square used for? A Punnett square is used as a probability or a prediction to say, okay, if we put these two parent flowers together and we had them had flower babies, seeds, well, what kind of flowers would we get? Maybe I don't want any white flowers. I want all pretty purple flowers. Or maybe I want a mix. Or maybe, how do I get white? The recessive trait. Well, let's find out. When we fill in a Punnett square, you bring the letter from the top down and the one that's on the left over. Okay, so in that case, that's a big P and another big P. Two big P's. That's the genotype that one of these boxes would have. We do the same thing, bring the top down and the left over. We repeat that process through all of these. And what we get are these four genotypes. Two of them are the same in this case. So we can use this to figure out what the flowers will look like. We have two big P's. Those are both dominant, so it's going to show the dominant trait, which is purple. We have one dominant, one recessive, like these other parents. That's also going to show the dominant purple. On this last one, we have two recessives. So there's no dominant purple to show. And that's going to show up as white. So this tool helps us predict what the offspring will look like of two parents if we know their genotypes. We can predict what the phenotype and genotype of their offspring, that's babies, will be. Okay, genotype are the letters, the alleles. Phenotype is the physical trait, like purple or white. Dominant is represented by a capital letter. It's one type of trait. The recessive is represented by a lowercase. That's the other trait. Recessive will only show if you have two lowercase. Dominant will show as long as you have just one dominant. So what I have here, I've lined them up. What's the percent of each genotype? 25 for two big P's, 50 for big P, little p, and 25 for two little p's. Okay, that's the percent of each genotype. What if I want to know the percent of the phenotype? Well, 75% are purple and 25 are white. At the bottom here are ratios. A lot of times they'll ask, what's the ratio? So it's 1 out of 4 to 2 out of 4 to 1 out of 4. 1 to 2 to 1 for the genotype. In the phenotype, we've got 3 purple to 1 white. That's all it is. Okay. Everything to do with Punnett Squares is going to be 25, 50, 75, or 100. Or 0. Sorry, or 0. Those are the only choices. So if it's got 33% wrong, 86% wrong, it's all with these kind of numbers. Okay. So again, Punnett Squares just show you what kind of offspring you can have from two parents. Now you know how to fill it in, what genotype, phenotype are, and what dominant and recessive are. The last thing I'll try to squeeze in here is if you have two of the same letter, like two big P's or two little p's, that's called homozygous. If you have a big P and a little p, so two different, that's heterozygous. I can try to zoom ahead and find that word written out for you. Oh, again, here's genotype and phenotype. Pause if you need to look at that. Let me find homozygous, heterozygous for you. That's right here. I'm just going to show them to you so you can see. If something's homozygous and it's got two capitals, we say homozygous dominant. This is heterozygous. It's also called a hybrid sometimes. Here's homozygous recessive, two little letters. Okay, so that goes over 6 really important words that you need to know. The 7th important word you need to know is allele. So each one of these little letters is an allele. Well, what is exactly an allele? We're not going to talk about that today, but each letter we shouldn't call it letter, we should call it allele. So that's what I'm going to try to do from now on, is refer to each individual letter as an allele. So let's just go back really fast just so you can see this one more time. Genotype, what genes it has, the alleles like big P and little p. And phenotype is the physical characteristics like purple or white flowers, brown hair, brown eyes, etc. Okay, so I hope that helps you understand how to fill in a Punnett square and what those words mean. All right, I'll see you soon.